Greetings. We are starting with the Cambridge IGCSE biology paper, which is paper 6, alternative to practicals. The paper code is 62, and this belongs to the October November 21 paper. The duration for this paper is 1 hour. This paper generally has a lot of diagrams, graphs, etc. So you need to be right at the top of the paper right from start. So, we are starting with the reading of the question. The student investigated the effect of temperature on the rate of respiration in a yeast cell. As we understand, respiration is a process as one of the major characteristics in the Mrs. Gren. Respiration formula, as we understand, it is glucose plus oxygen will provide carbon dioxide plus water, and in this process, ATP as an energy is liberated. Now, also we understand that respiration is a process which is dependent on enzymes. So, when we talk about enzymes, enzymes uh, have a lot of requirements. They require an optimum pH, that means the best at where they work. They also require what is called as the optimum temperature. Now, in this enzyme, we know that this enzyme has got what is called as an active site, which is a three-dimensional structure. In this active site, we know that when the substrate comes and fits inside in the optimum conditions like temperature, pH, etc., it can form an enzyme substrate complex. Why are we talking about enzymes over here? Because we said earlier that respiration is an enzyme dependent reaction, and for respiration to occur, it requires optimum temperature as one of the most important thing. So, moving ahead, we are talking about in this investigation, the gas was collected in the balloon that was attached to the test tube containing the yeast solution. So, what happens in this experiment is that when the yeast respires, it will release carbon dioxide. This carbon dioxide rises up in the balloon. As you can see now, the balloon is deflated. But as the gas will start to accumulate, you can see in the later part of the question that the gas has started to inflate and the carbon dioxide has started to take the space inside. Okay, so what are the steps that were taken care while doing this experiment? So, the student prepared a hot water bath and a cold water bath. So, here the student is trying to investigate the reaction of respiration under different temperatures. So, that is why hot and cold water bath. The student measured the temperature of the water in hot water bath and in the cold water bath. The yeast suspension was stirred with the glass rod. 25 cm cube of yeast suspension was put into each of these two test tubes. So the student measured the temperature of the water in the hot water bath and the cold water bath. Now, for maintaining the water bath, it is always better that we use what is called as the thermostatically controlled water bath. I will deal with that in the later part of the paper. But apart from that, you can also insert a thermometer over here and keep on monitoring if it is a warm water bath. Okay, then the yeast suspension was stirred with a glass rod. 25 cm cube of the yeast suspension was put in each of the two test tubes. The balloon was stretched over the top of each test tube. One test tube and the balloon was put in the hot water bath and the other was put in the cold water bath. The circumference of each of the balloon was measured by placing a piece of string around the widest part. So here you can see there is this balloon. They have placed a string here around this balloon. Once the inflation was done, or it was inflated, here you can see on the ruler, they will place this string and measure how much is it before and after the experiment, or what was the difference between the hot and the cold temperature. The circumference of each balloon was measured at a specific time interval, that is 0 minutes, 5, 10 and 15. Now, uh, the student recorded all these results in a table and the table was then prepared. So the question next says that prepare a table to record the results of to record the results shown. So when we are making a table, it is very important that we are going to make a proper table with all the lines and the requirements. We start with what is an independent variable on the left side and the dependent variable on the right side. So the independent variable was temperature, which was calculated, sorry, the individual temperature was time, which was 0, 5, 10, which was calculated in minutes. Okay, so I'm going to take the time on the left side, considering it as an independent variable, which was calculated in minutes. The dependent variable which we measured with the string was the circumference which was measured in millimeters. Two places where they were considered, or two conditions which they were considered, were cold and hot. We were looking at the time difference now. So the time difference was 0, 5, 10, 15. Now you need to plot the data which was present over there. So here on the top you can see 120, 125. So this is the data that you need to add in the table. 120, 125, 132 and 137. This was for the cold. And now the next table is, the details are for the hot temperature, which was 120, 135, 145 and 147. Right? So this is how you're going to get 3 marks for identifying the independent variable on the left side, dependent variable on the right side, and then putting in the details. This is how the 3 marks are distributed. Now, state a conclusion for the results shown. So here we are looking at the table where you can see that there is a difference in the temperatures. If you look at the cold, after 5 minutes it was 125, but in the hot it is 135. In the cold, after 10 minutes it is 132, but in the hot, after 10 minutes it is 145. So what is the conclusion that we come at? That the yeast cells respire rapidly at a higher temperature. This is what we call as a conclusion, means what you understand from this experiment. Other option is higher the volume of the gas, higher volume of gas of carbon dioxide was produced at higher temperature. But this I would rather call it as an observation. But when the question is asking you for conclusion, we will write this: yeast cells respire more rapidly at higher temperature. So this is what we are going to write. Now, suggest why it is important to stir the yeast solution in step 3. So here in step 3, we have seen that the yeast suspension was stirred with a glass rod. So why it is suspended? So that it is uniformly distributed and the reaction is occurring all throughout the suspension. So the answer is that the yeast cells should be uniformly distributed. Now, state two variables other than the strings that should be kept constant in this investigation. What do you mean by the term constant? That means it should be kept the same. Every time when we do an experiment, we will always repeat the experiment at least 2 more times, that is in all 3 times at least, so that we can find an average mean for this experiment. Or we can remove all the errors or the anomalies. Okay, so here two variables other than the stirring that should be kept constant. So it could be the type or the age of the yeast, concentration of the yeast solution, as in how much of yeast solution you've added in that 25 cm cube, which was not mentioned in this experiment, volume of the yeast suspension, type of the balloon, how big it is, small it is, how elastic it is, and all that, the time for observation, and the temperature of the water bath, as in the hot water bath, the temperature should be maintained at the same temperature every time you're going to repeat the experiment, or else it will affect the total results of the experiment. Okay, now balloons and strings were used to collect and measure the gas produced by the yeast cells. Suggest another more accurate method for collecting the measuring gas. Okay, so here you could use something called as the gas syringe, the bung or the delivery tube inverted measuring cylinder over water, or the downward displacement of water in the graduated container. So as we see Here, this is called as a glass bung. In this glass bung, as you can see that, suppose here the experiment takes place, the gas will travel down in this position. The gas here, when we are looking at carbon dioxide, starts accumulating and the liquid will be pushed down. The more the liquid goes down, at the end of the experiment, the reading can be taken. Then we are looking at this as a gas syringe. Okay, so here the syringe at the start of the experiment would be here. Once the reaction, like the yeast mixture here again will be reacting, the carbon dioxide gas rises and it will push the syringe, or that pressure, it will be pushing the part behind. Okay, so these are the different methods. Other method that we also spoke about was bung and delivery tube. So this is a beaker. Here you have taken a test tube where you are adding this yeast solution, and then you are talking about an another. So here you could take a conical flask also, and then the tube is inserted here, and then the gas will accumulate here. You can also see at the end of the experiment what is the final gas which is collected. State that the name of the indicator which should be used to show the gas produced is carbon dioxide. So as we know, here we are talking about the lime water Milky test. Okay, so you can see that here at the start of the experiment, this is the color of the yeast solution. When the carbon dioxide is released, it will turn the lime water milky. Or you could also use what is called as the hydrogen carbonate indicator. So hydrogen carbonate, if you see at atmospheric level, it is red in color. You can see this color at the start. As we are looking at that, the pH changes, the colors also change. Okay, and this is how, as the carbon dioxide concentration increases, okay, it will become more acidic because it dissolves in water to form what is called as carbonic. It will form carbonic acid. Okay, so you can see that the colors will start to go deeper, as in we are looking at orange, yellow, or if it is lower or in the alkaline, it starts to go from purple, magenta, etc. Okay, so this is the hydrogen carbonate indicator that is used very commonly for carbon dioxide test. Okay, further we are moving. The temperature of this of the water in the hot water bath and the cold water bath were measured again at the end of the investigation. Figure 1.4 is a diagram of the thermometer showing the temperature in two water bath. We are looking at the hot water bath, which is here around 28 degrees Celsius, and here we are looking at the cold water bath at 20 degrees Celsius. Okay, now they have already mentioned one table which is given over here about the temperature at step 2, which was for hot water 40 and then 20. So the change we have seen is 28 and 20. So they have asked you to calculate the change in the temperature. So obviously we are going to link 40 minus 28, which is going to be 12, and 20 at the start of the experiment and 20 at the end. So the result is going to be 0. So this is again for 2 marks that you need to do the simple subtraction. Describe how the method could be modified to prevent the change in the temperature of the water bath. Here we are saying you can use thermostatically controlled water bath. So as you can see, this is a thermostatically controlled water bath. There is a digital monitor over here. You can set the temperature to what you want, and the test tubes can be put inside this. Okay, so this is a thermostatically controlled water bath. This is a normal water bath, like I was telling you that the thermometer can be inserted here. Okay, this is cold water bath, this is a hot water bath. You can see the Bunsen burner has been ignited to raise the temperature of the water. You can also see on the thermometer the temperature is increasing, and here you can see there is no change in the temperature. This is also a thermometer which is called as a room thermometer, which will measure the temperature of the room. Okay. So coming back to the question, describe how the methods could be modified to prevent the change in the temperature. So you could use a thermostatically controlled water bath, which we just saw here. Then you could use a Bunsen burner and a thermometer. I showed you that diagram as well. Then you can use a Bunsen burner with a monitor the temperature. top up with hot water or cold water or ice. I showed you that method as well, or you could also insulate the container. Now, that was the end of the first question. The first question was 13 marks. Each question has got their marks at the end. You have a look at the question marks and then write the answers accordingly. Here, as you can see, this question is only for 1 mark. You need to write any one of the points provided. You need not write all the points provided, even though 3 lines are provided for this question. Moving ahead, we are looking at the diagram of this wind pollinated grass plant. You have to draw a large diagram of the flower. So as you can see, I have already drawn this for you. This is a 4 marks question. Now, things to be avoided or what you need to take care of. First thing that they will give you is for the magnification. So very important that you occupy 60 to 70% of the place provided. Then we are talking about the details. So as we can see here, 1, 2, 3, 4, 5, 6, 7, 8. Okay, so 8 lines are provided. You need to also do the same. So 1, 2, 3, 4, 5, 6, 7, 8. Okay, so this is what we are looking at the details. So for the details, the marks is provided. Now, another point which is provided is for outline. Now you need to be very careful, which I have deliberately done over here, is you are not supposed to do any overwriting. You are not supposed to break the line. It should be one proper solid line. Okay, so this is what we are talking about is the diagram, and other thing that is it should look like the diagram which is already provided. Okay, so this is the 4 marks for the diagram. Next question is for figure 2.2 is a photograph of a grass pollen grain. Measure the length of the line PQ. So the length of the line PQ, you will start measuring it at the top. Okay. You will start measuring it from here, and till this end point, the measurement what I have got is 62 millimeters. Now they have given you the formula for magnification, okay, and they have just asked you to give the answer in millimeters in 2 decimal point. Students, this particular thing is very important. If you don't follow the instructions as to what they have asked, you will lose your 1 mark. So whenever there is a space provided for the working, you have to do the working in that. Even if you use your mental maths or whatever, you still need to provide the working over here. So it is 62 was what the length I got. Okay, and the magnification is 15. Sorry. The magnification is 1500, which is already mentioned over here that it is already magnified 1500 times. So 62 divided by 1500, your answer would be 0.041333 and so on. But you are supposed to write it in 2 decimal places. So your answer is 0.04. So how is this 3 marks calculated? 1 mark for calculating the 62, that is the length, 1 mark for the proper calculation, and the third mark is for writing the correct answer with 2 decimal place in this dotted line. Always remember, always put your answers in the dotted line place provided to you. Now, scientists investigated the effect of wind speed on the average distance traveled by the single pollen grain, and groups of 5 pollen grains joined to form a clump. So here there is a single pollen grain reading and the 5 pollen grains together which were moving. Obviously 5 together would decrease the speed. So you can look at the observations over here. And when the wind speed was 0.2, in the single pollen grain it traveled 38, but in the group of 5 it traveled only 21 meters. Now, calculate the percentage decrease in the average speed distance traveled when the pollen grain fell as a clump compared to a single pollen grain at a wind speed of 0.6 meters per second. So here we are seeing this is 0.6 meters per second. The reading for single grain was 118 and the reading for 5 pollen grain was 55. Now, we are talking about the formula final minus initial upon initial. This is the formula for your percentage decrease. Okay, so and then you're going to multiply it into 100 because it is a percentage. So when we are looking at final was the difference that you're going to see, and then the initial, when you divide by it into 100, your answer is going to be minus 53. This is again your answer for 3 marks. Next, we are looking at plot a line graph on the grid of the data to include both sets of data and the key. So you are supposed to include the single pollen grain and the 5 pollen grain. Both of them have to be put in the same graph. You are looking at the wind speed meters per second as your independent variable, which needs to come on the x-axis, which you need to write here. Then you are looking at the average distance traveled by the pollen grain as your y-axis. This is also supposed to be written here. And then you're going to see the scale. Again, very important that you use more than half of the place provided here. And then you're going to put the keys. So for the single pollen grain, I have used this straight solid line. And for the 5 pollen grains, I have used a dotted line, and you can see the difference in the movement. This is what you need to plot. This is again a graph for 5 marks. Okay, x-axis plus the detail is 1 mark, y-axis plus the y-axis written over here with the detail is 1 mark, your scale carries 1 mark, okay, and then 1 for the single pollen grain and 1 mark for the 5 pollen grain. This is how the 5 marks are allotted. Now, use the graph to estimate the average distance traveled by a single pollen grain. So we are looking at this line as the single pollen grain at a wind speed of 0.5 per second. Now, when they say show on your graph, have you obtained your estimate? So 0.3, 0.4, and we are looking at 0.5. So you are going to start going from 0.5 on this line of single pollen grain. You will mark it here, and it is somewhere around 100 meters. Okay, this is how you're going to write your answer. This plotting has to be done on the graph when they say show on the graph. Okay. Successful pollination results in the fertilization and the production of the seeds. Describe how you could test the seed to show that they contain reducing sugars. For reducing sugars, we are going to use the Benedict's test. So Benedict's test, in the start, it is blue in color. But when we are going to heat it, or and as the sugar is present, it is going to turn from green to yellow to orange red to brick red. This is your final result. I have also given you the quantitative results over here. So what is the method for 2 marks? Add the Benedict solution or the reagent in a test tube. Heat it at 80 degrees Celsius for 2 to 3 minutes. Positive test would observe the color change. It is very important to write from blue to brick red, that is from initial to final. Okay, and this is how the positive test result can be inferred. Next question we are looking at. Plan an investigation to determine the effect of temperature on the rate of transpiration from the leaves. So, you may wish to include the apparatus listed and any other apparatus that you think is suitable for your plan. So, they have given you some ideas as to how you could do it. You could use leafy plants, electric balance, thermometer, string, and stop clock. Okay, so these are some um, options that they have provided. Now, how you can use the string and the electric balance? So you could weigh the leaf at the start of the experiment using an electric balance. Then you could hang it on a string and then use the same electric balance at the end of the experiment to see the difference in the observations. Okay, so this is how you're going to use one tripod stand over here. Okay, hang this over here with a string. Okay, again here, and then hang it over here, leave it for some time or maybe 2 to 3 hours, and then you can see the difference in the leaves. This thing that you can see is called as a photometer. Photometer measures the rate of transpiration. So here we are going to see that this is the apparatus. There is a beaker over here where they have inserted this bubble, which you can see here was at the start of the scale at the starting of the experiment. As the transpiration takes place through the stomata, the xylem will take in the water from this potometer, and the bubble will travel from the uh, start at here to a specific distance at the end of the experiment for whatever time you're going to observe this experiment. Okay, so this is how. So how I have written this experiment. The investigation is done using three different temperatures. Okay, that is 10 degrees Celsius, 25 degrees Celsius, and 40 degrees Celsius. Take three leaves which are weighed at the start of the experiment. So I'm using my electric balance. The leaf is then suspended on a string. So I am using the string which is kept at in a rooms with different temperatures. The leaves chosen will be of the same plant and the same surface area. These becomes your constant variable. So it is also controlled conditions like same humidity and light intensity, so that the temperature and all those, the humidity and all do not affect the rate of transpiration. We can use a thermometer to measure the temperature of the room. The leaf can be kept on the string for about 60 minutes, that is about an hour, after which we can use an electric balance to check the mass of the leaf after the experiment. The difference between the mass of the leaves will be measured and the experiment will be repeated at least 3 times to find the average mean. Always wear a lab coat and eye protection during the experiment. These are called as your safety precautions. Okay, so this is how an experiment for 6 marks is written. Next question we are looking at, humidity also affects the rate of transpiration from the leaf. Suggest how you could change the humidity of the leaves that are exposed to the transpiration investigation. So here what we looked at, this is called as a dehumidifier. Or if you want to add humidity to the room, you can use a beaker with water in this and keep it close to a vent or a fan, which will add humidity to the room. So these are the two methods which you could use for this question. This is dehumidifier again, and this is the way to increase the humidity. So to decrease the humidity, we could use a dehumidifier, and to increase the humidity, we could use bowl of water around the apparatus or place a plastic bag on the plant. So this is how the ATP paper is solved with all the details. Again, here this is an 1 mark question, so either one of the option you need to write. This question was for 27 marks. So 27 marks is the second question, and the first question was for that, and the total for this is 40 marks. This is how the ATP paper is attempted. For any further doubts, please do write in the comments. Do like and subscribe to my channel. Thank you.